All right, this video is going to be all about what antibodies do. So we've spent quite a bit of time talking about the need for antibodies. And so far, you know that antibodies are going to bind to the antigen, which is on the pathogen, and target it for destruction because we want it out of our system. But what exactly does the antibody do, right? Well, there are 5 main functions of antibodies. So you can see the list here, and we're going to show illustrations of these and talk about them in more detail. But I hope by the end of this video, you see just how important antibodies are and why it's important to have a strong immune system. So the first thing we'll talk about is neutralization, whether that be of exotoxins or pathogens. Opsonization, which is how we combat pathogens that have a capsule. Remember, with the capsule, they're trying to evade phagocytosis. Well, opsonization is going to be how we fight back to promote and enhance phagocytosis. Then complement fixation, which is going to be activating that classical complement pathway that we talked about with non-specific immunity. Then immobilization and agglutination. So let's go ahead and get started. So neutralization basically means you are neutralizing the threat, okay? So we're going to do that by preventing the threat from being able to carry out its role. So look at the top part of this picture. So there, the triangle is the exotoxin, and then you have that poor unsuspecting cell. And normally that exotoxin, which is a protein, it's going to act like an enzyme, it's going to wreak havoc on the cell and normally kill it. So that's what you can see happens there. Well, with antibodies, remember we have a specific term for antibodies that target toxins, and that is called an antitoxin. So once we've generated these antibodies, or as I should say, antitoxins, they are going to go and bind all those little exotoxins, which means the exotoxin is no longer free to harm the cell. Okay, so we've neutralized that exotoxin threat by binding to it, so we're preventing it from actually reaching its target, which is the host cell. And then it's the same idea for neutralizing other pathogens, specifically bacteria and viruses, because remember of that seven-step process for trying to cause disease in a host. Step 3, I think it was microbes trying to, attach to cells and perhaps invade into deeper tissues. And they did that either through the docking protein, if it was a virus, or with things like Fimbriae on bacteria. And so what these antibodies can do is bind to those pathogen structures like the docking protein or the Fimbriae, because if those attachment sites are blocked, then the pathogen can't attach to the cells and they can't get in, right? So again, neutralizing the threat by preventing it from accessing its actual target. All right, next up is opsonization. So as I just mentioned, many pathogens that we deal with have capsules because it's a sugary, sticky structure. Phagocytes have a hard time attaching to them. And if they can't attach, they can't ingest and digest, right? So that's a problem. And so what we do is we have antibodies that will go and coat the surface of that bacterial cell, for instance. So it coats that sticky capsule, which then allows the phagocyte to grab hold of those antibodies. Those antibodies act like little handles. The phagocyte can grab hold of the antibodies and conduct phagocytosis. Okay? All right, so we talked about the complement system in our non-specific immunity video series. And remember that the classical pathway was activated by antibody-antigen interaction. And so that's what you're seeing over here on the left. So you'll see the little antibodies that are kind of like a, I don't know, is that like a brownish, maroonishy weird color? So notice the antibody is bound to the antigen on the pathogen surface, right? That variable region. But notice what's happening at the constant region of that antibody. It has attached to C1, okay, which is part of the complement pathway. And so this antibody-antigen complex that has now attached itself to C1 will activate that classical pathway. And remember, there are 3 outcomes of complement: opsonization, which we just talked about, inflammation, and the membrane attack complex, which is MAC. And that leads to cytolysis, because remember, the MAC acts kind of like a pore that allows the cell to leak and eventually lyse. Okay, so by activating complement, there's 3 important outcomes that are going to happen. Next up is going to be immobilization. So remember, we talked about a strategy that some pathogens use to evade phagocytosis is to use their flagella and outrun the phagocytes. Because if the phagocytes can't catch them, they can't be ingested and digested and eliminated. So we don't want these flagellated organisms to be able to outrun our white blood cells. So some antibodies will attach to that flagella, which prevents them from operating properly, and it kind of immobilizes them. They become kind of stuck in place, and that way we can easily phagocytose them. Okay, it gives, kind of levels the playing field and allows our white blood cells to catch up and do their job and eliminate this bacterial pathogen. All right, last up is going to be agglutination. So this is the last function of our antibodies. And so what I want you to look at here is the green circle is the pathogen, and you see that it's little antigen and epitope there. And look at what is happening with each antibody. Do you see how each antibody is attached to 2 different pathogens, right? And so if you look kind of at the far right, you can see that that first antibody is holding on to 2 different pathogens. Then if you look up a little bit, that same pathogen is being held onto by a different antibody. Okay, so essentially what's happening here is the antibodies are rounding up all the pathogens and kind of localizing them to one spot. That way, a phagocyte can come and just phagocytize all of them at once rather than chasing down each individual little pathogen. Okay, so agglutination is like a word for clumping. Okay, so I use the term round up as a way of like saying we're wrangling all of these pathogens together and kind of holding this clump of them in one spot so a phagocyte can come and eliminate it. Just makes phagocytosis more efficient. All right, y'all, that is going to be the end of this video. Next, we're going to be talking about our secondary immune response. Let me know if you have any questions.